Hi, dear children. It's been a while. I thought we would complete the entire section on nervous coordination by looking at questions. So what I've done is that I have chosen quite a few questions from past year question papers, including some trial examinations from the other provinces. And as we go through them, I'll be pointing out various things, including how you should be answering questions, how important it is to read questions before you attempt to answer them, and be very guarded because many learners presume that the same information will not be tested. Of course, the same questions will not come out, but the same information is tested. So if you know your information very, very well, and you know the skills that govern answering questions, there should be no problem in doing very well in your life science paper. So, here we go. Let's begin. We're first looking at a question from June 2019, Eastern Cape, my darlings. And let me just make it a bit bigger for you. It says the brain acts as the main coordinated center. It's supposed to be coordinating, my darling. Center for nervous activity. It receives information, interprets it, and responds accordingly. So that's what the brain does, isn't that so? It receives information, interprets the information, and provides an appropriate response. Just in case they ask you a general function of the brain, and that's a beautiful way of stating it. Anyway, let's read through. So let's be a little naughty and just put the labels here before we read through, my darlings, so that, in fact, you do the labels before I'm going to put them in, so that you can check how much you have learned from nervous coordination. How's that? Okay, so let's begin with this one. Yep, you're right, cerebrum. Okay, let's go to B. Yes, right again, cerebellum. Let's go C, darlings. C, it's quite high up there. Of course, this would do your medulla oblongata. Now, if you're not sure, of course, your questions will direct you as to what C could possibly be. Oh, it's not medulla, it is medulla. And remember, when you're learning, have some fun with yourself. It's okay to be wrong, as long as you're not wrong all the time. And of course, what would that be? You'll see that your pituitary is attached to it, isn't that so? This is your pituitary, so that would be your hypothalamus. Once again, if you're not sure, be guided by your questions. Okay, let's just run through how we remembered the function cerebrum. Jim the 4th, cerebellum, BC, hypothalamus, taste P, and then the medulla oblongata, ICC. Okay, so if you don't know what those mnemonics stand for, go back to the previous lessons, and of course you'd be able to find out an easy way to remember this. Okay, darlings, give the letter only of the part of the brain that controls the following: breathing. Then you know for sure that C is medulla oblongata, isn't it so? Because medulla oblongata is for involuntary functions, and breathing is an involuntary action, like heartbeat is involuntary. You do not have conscious control over it. You can't tell your heart what to do. Okay? Memory, of course, it's a higher thought process, isn't that so? Judgment, intelligence, memory is A, cerebrum. The part labeled A is made up of two similar halves, the right and the left. How are these two halves attached to each other? Of course, my darling, by this band of nerve fibers here, isn't that so? That's the corpus callosum. State one way in which the brain is protected. Of course, there's quite a few ways, isn't it? So you've got your meninges, which are the membrane around it. You also have your skull. Which part of your skull? Don't say. I love for you to say cranium. And of course, you also got your cerebrospinal fluid, which is found between the meninges, CSF. State 2 reasons why humans need a nervous system. There we go, boo-boos. Why do you need a nervous system? It receives information, interprets it, and responds accordingly. And this is the way they put it, so that you can react to stimuli, you can sense stimuli, you can sense your stimuli, or detect changes, which is the same thing as sense stimuli, both in the external and internal environment, as well as, my darlings, coordinating the various activities of the body. Sorry, babies, to detect and to react and to coordinate the various activities of the body. Here you go. I didn't know this question was coming up, but look at it, my dolls. To detect changes in the environment and to react to these changes. It's exactly what they're saying over here. Go back here. See? It receives information, interprets it, and responds accordingly. It causes you to react. If it receives information, of course, it's your receptors and your sensory neurons bringing the information up. So that is how you detect stimuli, isn't it? So instead of saying detect changes, you could have said detect stimuli. How's that so far? Okay, so there's checking if you're right. Okay, we've done well. Okay, so what I'm going to go now and take you down to a question where you find that, my darlings, this is another easy one here before I go to the one that I want to point you to in developing skills for answering questions. Once again, boo-boos, A now. Oh, we're lucky because we got A and B. Because A would be spinal cord, B would be medulla oblongata, C would be, there we go, that band of fibers there, isn't that so? Corpus callosum. D is your cerebrum, E is your cerebellum. We've done that. Write down the letter and name of the part. So be careful, letter first and name that has a center for interpreting taste. Interpreting taste, this one here interprets all your sensations, isn't it so? D, cerebrum. I remember in one question they said, write down the name and letter of the part that would receive impulses from your heart. Give the letter and name of the part that will receive impulses from your heart. Now your heart, my darlings, you cannot have conscious control over it, isn't that so? Are you with me? So therefore, if it's an involuntary action, babies, it will be B, medulla oblongata. So that was a lovely one, which receives information from your heart. Regulates the heart rate. See, it's another way. So another question said, receives information from your heart. Here they're saying regulating the heart rate, same thing. B, medulla oblongata. And give the part that is responsible for motor coordination. Motor is to do with your muscles. Got nothing to do with the car that daddy drives. Okay? Motor coordination, remember I taught you, cerebrum is a shorter word. It controls voluntary actions. Cerebellum is a longer word, coordinates. So which one is responsible for coordinating? E, cerebellum. Let's check if we're right, dollies. There we go. Okay, now I'm going down to the question which asks you these functions, but in a different way. Okay? Ask you these functions. Sorry about running through this with you, babies. All right, you'll notice I've got a variety of questions. Here we go. Let's look at this one. Asks you the functions of the brain, but in a different way. Now, remember, my darlings, just asking you the function is a level 1 question. But I can change it to a level 2 or a level 3 question by saying, predict and explain what would happen if this part of the brain was damaged. So what are they asking you? They're asking you, what is the function of this part of the brain? Because if this part of the brain was damaged, then these functions will not take place. So papas, when you see the word predict or what would be the consequences, no, they're just asking you the function in another way. So please do not hyperventilate when you see these things. In 2002, link between concussion and brain damage. Concussion is when you get a direct hit on your head, or if you're involved in a car accident and your head is thrown, backwards and forwards in a very harsh movement. So it's actually like your brain moves inside of your cranium. So that is regarded as a concussion. In 2002, a former American football player was found dead in his truck. The doctor had handled the autopsy. Autopsy is when you find out what is the cause of death. Autopsy, of course, is done when someone is dead. Discovered that the football player had severe brain damage and that his death was caused by repeated blows to his head. If you've ever watched American football, my darlings, you would know why this is possible. This could also be possible during rugby as well, severe contact sports, or during boxing. That his death was caused by repeated blows to the head, or what they call repeated concussions. He called this disorder chronic traumatic encephalopathy, CTE. A more recent study was conducted which involved the brain of 165 people to try to make it a little reliable. They should have done a little more, but 165 is better than 2. Reliability of study. Who played football at high school, college, or professional level. The study found evidence of CTE in 131 of those brains. Dreadful, isn't that so, darlings? The part of the brain affected by CTE is the cerebrum. State 2 possible symptoms of this disorder. My God, and you're going to say, oh, my ma'am didn't teach me this. But they did teach you the functions. Because if CTE affects the cerebrum, that means the cerebrum is no more functioning as it should. So if it's no more functioning as it should, means it's not doing what it should do. So what were the functions of the cerebrum? We said, my darlings, you remember it as Jim the Fourth, isn't it so? If you remember it as Jim the Fourth, then you would know that the cerebrum is responsible for your higher thought processes like judgment, intelligence, memory. They could lose their memory. Are you with me? I stands for, it receives and interprets impulses from your sense organs. So you would have difficulty maybe in hearing, difficulty maybe in seeing. That, let's see which part of the cerebrum and how they involve these functions in saying to you what are the 2 possible symptoms. And V would be, you would lose your voluntary actions like walking, talking, etc. So let's see how they answer this here, my darlings, and then you'd be having an idea. Loss of higher thought processes, loss of one or more of the senses, loss of voluntary actions, and look at it, babies. It's all the functions, but you're not going to say, oh, the cerebrum is responsible for higher thought processes. You're not going to say it. You will say there will be a loss of higher thought processes. There'll be a loss of one or more of the senses. There'll be a loss of voluntary actions. There we go. So they're just asking you the function, but in a different form. So don't ever get worried, okay? Don't ever get worried. They're just asking you the functions in a different form. State 1 way in which the brain is protected. Now we're good. We know this one, isn't that so? We've come across it before. And if we come across it before, there we go. The cranium, the meninges, the cerebrospinal fluid. Explain why CTE does not usually affect essential life processes such as breathing and heart rate. Now we know that breathing and heart rate is controlled by the medulla oblongata. Now what do they say CTE affects? The cerebrum. It's because CTE affects the cerebrum that you will not have breathing and heart rate affected, because breathing and heart rate is controlled by the medulla oblongata. Let's see how they answer. CTE mainly affects the cerebrum, therefore the medulla oblongata, which controls breathing and heart rate, is generally not damaged. Exactly what we said. How's that, my darling? I think we've done well in that day. Okay, babies? Now, let's go to a question on the, here we go, this one here, isn't it so, papas? On the reflex action. Reflex action. Let's just share how we remembered the reflex arc. How did we remember the reflex arc? We remember it by the mnemonic. Let's go. Jim. Oh, no, it's not Jim. I'm still thinking of Jim. Roy. Stop calling me. Isn't that so? But me separate. So the stimulus is received by the receptor R and an impulse is created. So here, the impulse will be created. The receptor would be the heat receptor in your skin. S, the sensory neuron will now take, let me just do the spotlight for you, boo-boos. Okay. The receptor will now take this impulse up to the spinal cord using the dorsal root of the spinal nerve. This part here, darlings, although we don't focus so much, notice it's a fat part of this dorsal root. This is because all the cell bodies of sensory neurons lie here, and you need to create the space. So a space where you have a collection of cell bodies is referred to as a ganglion. Let us just draw this ganglion. A place where you have a selection of cell bodies is referred to as a ganglion. We generally don't focus, but we want to cover all our bases, okay? Ganglion. So anyway, so let's go back to our mnemonic and let's get our spotlight again. So then the sensory neuron, notice the sensory neuron comes right up from the receptor, up the dorsal root, and comes to the spinal cord. In the spinal cord, notice there's a dotted line here indicating a synapse, a microscopic gap between the sensory neuron and the next neuron, which is now our connector neuron. And then the connector neuron makes a synaptic contact, yet another synapse, with the motor neuron, which motors down the ventral root and finds its way, here we go, finds its way to the effector. And then the effector will respond, bringing about a response to that stimulus. By the way, my darlings, this is an external stimulus, okay? So I just want to refer you to something that I didn't concentrate on in your nervous coordination, videos, where I did not speak to you about the autonomic nervous system and the somatic nervous system, okay? So just let me just tell you something about it, okay? You've got your central nervous system, and then you have what is your peripheral nervous system, okay? Central nervous system, my darlings, is the brain and the spinal cord. Your peripheral nervous system is made up of, let me just try and draw this for you. Why are you not drawing? Okay. Is made up of two divisions, okay? So your peripheral nerve, oh, by the way, if you're confused, of course you should be at this point. If your brain and spinal cord make up your central nervous system, what is your peripheral nervous system is made up of all the nerves that come out from the brain and the nerves that come from the spinal cord. You don't have to know how many nerves, but actually there are 12 from your brain, known as cranial nerves, 31 pairs of nerves from your spinal cord. So anyway, those nerves will now supply different parts of the body. And that makes up your peripheral nervous system, which is divided into the somatic nervous system. Now, somatic nervous system, somatic nervous system is that part of the nervous system that generally responds to external stimuli. Remember, a stimulus is a change in the environment. External stimulus would be something outside of you, like the sound of music, like the smell of roast chicken, like the heat of a candle on your skin. Those are external stimuli. So the somatic nervous system is involved in this particular pathway or the reflex arc that you see, responding to an external stimulus. Then, my darlings, responding to internal stimuli. That means what is happening inside of your body and what you are not consciously aware of. Your body is constantly working so hard to keep you alive, babies. Working so hard to keep you alive. Yet most of us ignore this wonderful bodies that we have. And we only focus on all our external manifestations and get so depressed if there's even a little pimple on our forehead. Not forgetting that all the things that are happening inside of us are working for us to stay alive and know that there's a chance of getting rid of that pimple. Okay, loves. So the autonomic nervous system is designed to make you react to your internal stimuli. Now, what are some of your internal stimuli, darlings? Internal stimuli are like the level of your CO2. Internal stimuli are stimuli which are changes which are occurring inside of your body. Okay, those are internal stimuli. So your autonomic nervous system, my darlings, is made up of nerves which supply each organ of your body. Each organ of your body is supplied by nerves. Now these nerves, darlings, these nerves that will supply each organ are nerves that are antagonistic. In other words, they work opposite to each other. They work opposite to each other. Now, the first one is called a parasympathetic nerve. Parasympathetic, so that a heart will receive a parasympathetic nerve. Parasympathetic nerve. Okay, the heart would receive a parasympathetic nerve. There we go. And the heart will also receive, darlings, remember it receives, got two nerves, isn't that so? It'll also receive a sympathetic nerve. So each organ in your body receives sympathetic. I'm not known to type brilliantly. Of course, you would have seen that by now. Oh my goodness. I'm not even known to even spell properly. So your autonomic nervous system is made up of each organ in your body receiving two nerves. One called the parasympathetic nerve, one called the sympathetic. Let's take the organ to be your heart. Now, whenever you sympathize with somebody, it's like, oh God, you're calming a person down, don't be upset, etc. But that's not what the sympathetic nerve does. A sympathetic nerve increases the activity of the organ. So your heart will start beating faster. And the parasympathetic will decrease the activity of the organ. Okay, my darling? So if they ever ask you about the autonomic nervous system, it deals with the body recognizing the changes in your environment inside of you and how your body copes with these changes without you even knowing it. And it's made up of each organ in your body supplied by two nerves, the parasympathetic and the sympathetic. The sympathetic speeds up the activity of the organ. Parasympathetic would now slow down the activity. Okay, so which of these systems are involved in this reflex arc, where it helps you to respond to changes in the environment by first sensing the change in the environment, that is your external environment. It is your somatic nervous system. Good? Okay, so let's go forward. Great, my darlings. Good. Okay, let's see now what the questions are. Identify part A. Of course, that's the ganglion I told you about. B, we said is a synapse. C, you know that, connecting neuron. D, lovely, you say it motors down, so it's a motor neuron. And E is the effector. Specifically here, the effector is a muscle. So once again, if they say predict what would happen if this neuron was severed, severed means cut. It's not severed, it's severed, S-E-V-E-R-E-D. If this neuron was severed, now they're actually asking you, do you know the function of the motor neuron? So if it was severed here, Papas, let me just sever it, okay? If it was severed, let me just draw a line, okay? That means it was cut, if it was cut here, okay? If it was severed, means this impulse now would stop here. Means no impulse will travel down to your muscle. The muscle will not respond to a stimulus. In other words, my children, you'd be able to sense the stimulus, but you will not be able to react to it. So they're just asking you once again the function in a different way. That's what they're doing. They're asking you the function in a different way. So never get scared. Just don't get scared about these things. They're just changing the action word in the question so that they would satisfy the different levels that is required in your paper. E we've done. State one function of each part. What did they say, babies? I didn't get it. B and C. B, papas, synapse. It allows the unidirectional flow of the impulse. The impulse must travel from the sensory neuron to the connecting neuron. So it must know it must go from there to there. Unidirectional flow of impulse. That's a function of a synapse. And C, my darlings, oh, of course, it connects the sensory neuron with the motor neuron. And you can go a little further because you know so much now via synaptic contacts. Remember, neurons do not touch each other. Explain why the brain is not involved in reflex action shown above. So, darlings, that means from this receptor over here, the impulse is created, it does not go to the brain because the spinal cord is faster to reach. The brain will take a longer pathway to reach. So if you reach the spinal cord faster, you'd be able to react faster. That's very important because a reflex action is a rapid action. And a rapid action is only possible because the impulse travels a shorter distance rather than going to the brain and then coming down. So therefore, it allows you to respond rapidly to prevent injury to your body. Make sense? Oh, by the way, you must know that once a reflex action is over, then an impulse is sent to your brain to make you aware of what is happening. Remember, a reflex action is a rapid involuntary response to a stimulus received by a receptor or a sense organ and to bring about an action. So you do a reflex action without thinking. After the reflex action is over, then your brain becomes aware and you think, oh my God, it's burning. Because burning is a sensation, and sensation is only interpreted by the brain, not felt. It's only interpreted by the brain. So only when your cerebrum interprets this pain, you have that sensation of pain. Okay? So, explain what the effect will be in the action shown in the diagram above if a person is suffering from multiple sclerosis. Now, you've done multiple sclerosis, a level 1 question where your myelin sheath of your neurons break down. So in other words, here they're asking you, do you know the function of a myelin sheath? Because if you know the function of a myelin sheath, then you would know what would happen in a person suffering from multiple sclerosis. Now, you know, from theory, from a level 1 question now, papas, you know what the myelin sheath does. But if they ask you the function of the myelin sheath, then they're asking you a level 1 question. But if they say explain, then they're shifting the level 1 question to a level 2 question. Are you with me? Satisfying that grid that we have to satisfy when setting a paper. So, what would be the effect? Oh, by the way, let's just look at this one here, darlings. Explain why the brain is not involved. Spinal cord shortens the reaction time by sending impulses directly to the effector. If the brain is involved, it delays the reaction, causing injury or death. Same thing we said. So, once again,
what effect will be in the action shown? Remember, the action is a reflex action, which needs to happen very fast. So what would happen if the myelin sheet is broken down? The myelin sheet, they didn't tell you the myelin sheet is broken down. So you had to first say that in multiple sclerosis, the myelin sheet is damaged. This will now slow down the transmission of impulses. And if you slow down the transmission of impulses, it will result in a slower reaction time, which might lead to injury or death, depending on what the stimulus was. In this case, most probably injury. I haven't heard of anybody dying from a candle burn. Okay, loves? Not bad, isn't it? Explain the effect on the body if part label D is severed. Once again now, there you go. You could have even predicted that question. That's how good you must be at this stage. So, this can now be classified as a level 3 or level 4 question. 4 marks. The question is, how do you get these 4 marks? You always answer to the mark scheme. Never, never, never in life sciences do we give 2 marks for any particular question unless it is multiple choice. If we say explain something, we give 2 marks, you must have at least 2 parts to your answer. If we say explain, we give you 3 marks, you must have 3 parts to your answer. Okay? So, like this one here, 1, 2, 3 parts. Are you with me? Here, 2 parts, you can choose any 2 there. So, your motor neuron, the person will be able to feel the pain because the sensory neuron is able to transmit the impulses to the brain. Are you with me, Papa? The sensory neuron, you remember I told you from here the impulse will go to the brain. It's actually from the sensory neuron after the reflex action happens that it goes to the brain. Now, that is severed, so that doesn't stop the impulse going from the sensory neuron to the brain. Okay, so look at it, Papas, again. The person will be able to feel the pain because the sensory neuron transmits the impulse to the brain. Okay. However, no reaction will take place because of the fact that no impulse will reach the effector due to the damage of the motor neuron. So 2 parts to the answer. You'll be able to feel the pain because the sensory neuron carries the impulses to the brain, 2 marks. And then there will be no reaction to the stimulus as you find that no impulse will reach the effector. Explain the effect. I would have thought that you would also cause damage to the part of your body. But notice here, they haven't even included it in your memo. If I've ever seen something that is so fluid, it is your memo in life sciences. Whilst for this year, this would stand as a memo, next year, this would not stand as the absolute memo. So my advice to you would be, write as much as you can, especially in a 4-mark question, just to cover all your bases. Great, my darlings. I think that's sufficient for your reflex action. Let's do a little bit on the eye. Okay. Remember, we learned about the eye, the structure, the pupillary mechanism where the size of your pupil changes. We learned about vision, the pathway taken by light through the eye, and we learned about accommodation, which is a change in the shape of the lens for near and distant vision. Great. So now let's look at this one. The diagram below shows you 2 eyes, X and Y, focused on objects represented by arrows. So the objects here are these arrows. They could have been something that you're looking at, maybe a pole. Okay, Papas. So at different distances from the eye. Oh, once they talk about different distances from the eye, you know they're going to be referring to accommodation. Where, just let's recap accommodation, where the convexity of the lens changes for near and distant vision. So if it's near vision, we said, there was a lovely mnemonic by which we remembered near vision. For that mnemonic, go back to the lesson on the different functions of the eye. But we said for near vision, your lens becomes more biconvex, fatty, really fat lens it looks like. And for distant vision, it becomes less biconvex or flattened or thinner. Remember that? I always teach children to remember it by, if you're doing long distance running every day, like I don't, and you'll become thin, like I'm not. Okay, so for distant vision, your lens becomes flattened, or let's be scientific, less biconvex. So once again, let's focus. The diagram shows two eyes, X and Y, focused on objects. Someone said to me, ma'am, please use the spotlight. On objects represented by arrows at different distances from the eye. Objects A and C were 3 meters from the eye. Oh, that's another thing you gotta know. Near vision is less than 6 meters from the eye. Distant vision is more than 6 meters from the eye. So A and C were 3 meters from the eye. That means near vision. B and D were 8 meters from the eye. Distant vision. Okay, good. Diagrams are not drawn to scale, that's okay. Let me make this a little smaller so that we can focus on both the eyes. Great. If you look at eye X, oh, by the way, you must know that if you're looking at something, an inverted image is formed on your retina. And when impulses go to your cerebrum, then the image is now flipped around so you can see it the way it appears to you. Very importantly, my darling, this happens so fast because you're not looking at the world upside down. You always seeing somebody straight up unless they're upside down. Okay? So whatever object that you have, form, the image of the object formed in the retina must be the opposite of this one. So object A, is it opposite here? Yes. Object B is, oh, it could be A or B because both of them are opposite. But 3 meters from the eye, my darlings, means it's close to the eye. Now, if it's close to the eye, your lens becomes more biconvex. So just look at both these here. Which is more biconvex? This one. Are you with me? So therefore, A would not apply to this one. 8 meters from the eye, your lens becomes flattened, so it's object B. So for eye X, it's looking at object B. How's that? So let's look at eye Y. Remember, an upside down image is formed on your retina. Oh, so both of them, my darlings, so C and D, it could be C or D, but you've got a fat lens. So fat or more biconvex for near vision. So this is 3 meters from the eye. I don't even have to go there. So it's object C. So for eye X, it's object B. For eye Y, it's object C. Near vision. This is 3 meters from the eye. That's 8 meters. So if you're looking at object C, then I know it's object C because your lens is now more biconvex. Know the logic. So we know that IY is focused on C, IX is focused on B. Name the eye defect which results from the inability of IY to focus on object D. Object D. So IY, my darling, object D is 8 meters from the eye. So if you can't see things which are far away from you, that means you are short-sighted. Are you with me? So name the eye defect, short-sightedness. Are you with me? And short-sightedness, by the way, my darlings, how would you correct? Name the type of lens. It is biconcave lens. Okay? Long-sightedness is biconvex lens. Learn that off by heart. By the way, boo-boos, another way of showing short-sightedness is that in one question paper, I saw that they drew a long eyeball and a short eyeball. A person who's short-sighted normally has an eyeball which is longer than normal. So it wouldn't be like rounded like this, papas. It'll be an eyeball. Oh, goodness me, I wonder if I can do this. it'll be an eyeball which is longer than normal. So your eyeball will be as long as that, like that. Not long like that, but I'm sure you get the gist. Okay, so in another question paper, they drew a long eyeball and they drew a short eyeball. So this one here, darlings, if you say that is long, and for your long-sightedness, you'll have an eyeball that is shorter than normal. Shorter than normal. Okay, so remember that. Okay, so there are reasons why you're short-sighted and long-sighted. The easy way in which I remember it, my darlings, is that short-sighted, take the opposite of short, long eyeball. Are you with me? And then the other one is that your lens fails to become less biconvex, because to see distant object, your lens must become less biconvex, like that. So because your lens cannot become less biconvex, you cannot see a distant object. Because your eyeball is longer than normal, you cannot see a distant object. Here, you cannot see an object close to you because your eyeball is shorter than normal. So this means you're long-sighted, nonos. Take the opposite of long would be short. Because your eyeball is shorter than normal, and then, my darlings, the second one would be because your lens cannot become more biconvex. I'm just exaggerating so that you get the picture. I do hope you get the picture. Okay, quite a few things we learned here. Okay, let's go now. Name and describe the process that allows IY to form a clear image on the retina. IY, my darling, name and describe the process. Of course, the process is accommodation. Okay, so IY is focusing on object C. So you got to describe all the changes that occur for accommodation for near vision. And you remember that by the mnemonic CC. Let's go to that mnemonic again, just to recapture what we learned. Okay, so for near vision, if you know the one for near vision, then the opposite will be for distant vision. So here they're asking you, because this one is focusing on object C, that's near vision. So it is C, C, S, L, S, and L, B. Those are the ways in which you remember it. So here, your ciliary muscles contract, your suspensory ligaments slacken, tension on the lens decreases. So therefore, the lens becomes more biconvex. So because you're not pulling on the lens bulges more or becomes more biconvex, increasing the refractive power of the lens. You must say that. Increasing the refractive power of the lens, causing a clear image to be formed on the retina. Great. Okay, so please, there are certain things that are non-negotiable that you've got to learn off by heart. So there we go, ciliary muscles contract. Something curious, you know, and I told you it's biconvex and biconcave lenses as the answer. And some memos will give you as concave. So my darlings, as I said to you, that fluid memo just surprises us. So one of the recent memos, they said instead of saying concave lenses, say biconcave lenses. Instead of saying convex lenses to correct, you say biconvex. Of course, these lenses are forming part of your spectacles. So it's spectacles with biconcave lenses or spectacles with concave lenses. Spectacles are the glasses you wear, by the way. Many people just say glasses and they want to know what on earth is spectacles, ma'am. Accommodation, ciliary, you had to say accommodation takes place, 1 mark. It's not just compulsory mark. If you didn't use the word accommodation, you would have, and you got all of these right, you'd have just got 4. 4 out of 5. You just lose a mark unnecessarily. As I said to you, when you have something that is given a lot of marks, try and cover all your bases. Remember, in your answers that you are giving, you actually teaching the person who is reading. Of course, the person who's reading is the life science teacher, but it must be like a recipe that is to be followed. So go back and read your answer and say, listen, would I know what this person is talking about? Okay, so be as clear as possible. Accommodation is the process. Ciliary muscles contract, suspensory ligaments slacken, tension on lens decreases, lens becomes more, see, I said to you, biconvex, convex, refractive power of lens increase, clear image forms on the retina. Ooh, I would have got full marks for that. Okay, darlings, so that is the eye. Just don't want to go to the ears yet. I want to focus on another question on the eye, where they normally talk about the diameter of the lens, the convexity of the lens or the diameter of the pupil. I'll come back to the visual defects one. I just want to go back to this. There we go, the diameter of the pupil one. Okay, let's look at this question. Please, in life sciences, I think with every paper, reading time is reading time. Take your time to read. Read and read again. Don't use your time to answer your multiple choice questions. You will take the time to read the questions in section B and section C. Multiple choice questions you'll have to read as you're going through it. But what happens is that, my darlings, because you're trying to answer your multiple choice questions, you cannot judge how much of time you should be spending on each of the other questions and in your essay. So if you'd want to even answer your essay first, begin with that, as long as you number correctly. That is very important, just in case you think you'd forget your mnemonics, or because essays are generally level 1 questions. Level 1 means you should have known the knowledge. Okay, so if you choose to answer your essay first, great. Then you can go to your other questions. But very importantly, the mistake children make is that they go through the multiple choice questions in reading time and answer every multiple choice. God, you're going to do that anyway. You need the time to read all these wordy questions. Very verbose, some of them are. So you got to read and try and focus on what is being asked of you. So read the preamble. This here is known as your preamble, my darling. The information that is given before. Let's do our spotlight again. The information that is given before the question itself. Okay? The iris of the eye has the ability to regulate the amount of light entering the eye in order to prevent damage to the eye. Yes, of course, because if too much light enters the eye, the retina is damaged. It does so by regulating the size of the pupil when it is exposed to varying light intensities. We know that when it is bright, the pupil becomes small. When it is dim, the pupil becomes larger. I like to use the word constrict and dilate. So when there's bright light, the pupil constricts, becomes smaller. When it is dim light, the pupil becomes wider or dilates. Now, what has that got to do with light intensities, you may ask? So when the light is bright, then the pupil becomes smaller. When the light is dim, the pupil becomes larger. Sometimes you don't need varying light intensities. All you need is varying distances. No. So what do you mean by varying distances, my children? So varying distances would mean, for example, let me just do this. Varying distances would mean, for example, I've got my light there. Are you with me? And I am looking, my eye is here. Oh, goodness, let me just do this eye. Okay. Okay, there's my eye. So the distance from there to there. Then, after a while, I will take that. Let me undo that distance, my babies. Okay. So that's where it was. And then what I'll do is that I'll move it closer. Oh. So now it's closer. And if it's closer, that means that light would appear brighter to me. and then my pupil would be small. So instead of changing the light intensity, I can just change the distance of the light from the eye. Okay, be aware of something like that. Okay, good. The process was as follows. A person in a darkened room was asked to cover one eye. So very importantly, my darlings, when you're doing any investigation, there must be only one factor that is varied. That is known as your independent variable. Everything else must be identical. Otherwise, your investigation would not be valid. Every investigation must be valid for it to be recognized, for the results to be recognized. And for it to be valid, once again, only one factor must vary. The factor that will now change depending on the factor that you vary is called the dependent variable. In other words, when I change the light, what change would I see in the eye would be my result or my dependent variable. Okay? In a table, if the table is like so, my darlings, if the table is like this, put horizontally, then the upper one is normally your independent factor variable, and the lower one is your dependent. So this would be your, what did I say? Normally. Are you with me? Normally. And that would be your independent, that will be your dependent. You know what I'm talking about just now when I stress the word normally, okay? Or your table can be drawn this way. Many of you are used to your table being drawn, yeah. Oh, not text, sorry, babies. Many of you are used to your table being drawn there. Vertically, not horizontally. So you have a table like so. And in something like this here, whatever is here on the left-hand side, whatever's on the left-hand side is the independent variable. Whatever's on the right-hand side is the dependent variable. Okay. Now, if you're confused about which is the independent and dependent, always go back to what they wanted you to investigate. Because if you check what they wanted you to investigate, you can work out your independent and dependent variable. Let's look at what they wanted you to investigate. Scientists did an investigation to determine the effect of dim light on the diameter of the pupil. Whatever you want to see the effect of, that is your independent variable. So go down here when I say normally what is there. This is just the time intervals. That doesn't become an independent variable as much as the effect of the light. The effect of the dim light in this case, they said, that will be your independent variable. That what does it have an effect on? Diameter of the pupil. So when you read that sentence, what the scientists wanted to investigate, you can work out the independent and dependent variable. So if I say, scientists did an investigation to determine the effect of temperature on the rate of photosynthesis. So temperature becomes my independent variable. Rate of photosynthesis becomes my dependent variable. But it's so hard to measure rate of photosynthesis. So you would find that they will ask you, which is the dependent variable and how is it measured? If they do that, then you know it is difficult to just measure the rate of respiration or the rate of photosynthesis. So there must be a physical way of measuring it. No? So whenever it's a dependent variable, there must be a physical way. You must be able to see something. You must be able to see something to know what the results are. Okay? So once again, whatever they want to determine the effect of, that becomes the independent variable. Whatever it has an effect on, that's the dependent variable. So if I want to see the effect of dim light on the diameter of the pupil, I hate to say dim light, but the light itself becomes the independent variable. The diameter of the pupil becomes the dependent variable. So which are the things that he will keep identical? The one thing that he's going to change is that light. The one thing he's going to change is that light. So how does he change the light? We'll see just now. The procedure was as follows. A person in a darkened room was asked to cover one eye. Now, whatever eye that person covers, you will use the eye that is uncovered for all your investigations. You will check the object with that uncovered eye. Don't suddenly switch and say, oh, let me put the patch on the other eye. No, my darlings. So to keep it valid, you will use only the same eye to look at the light. A dim electric bulb was positioned at varying distances from the person. Positioned at varying distances from the person, was switched on at 1 minute intervals. Of course, the 1 minute intervals, you can see it here, appearing in the place that we'd have our independent variable. But this, my darlings, is like saying what happens in test tube 1, what happens in test tube 2, what happens in test tube 3. You can't say test tube 1, 2, and 3 are the independent variables. So once again, if you're ever confused, say, which is the independent variable, you can't say time interval. It's just giving you information as to what was the diameter of the pupil at these times. So they didn't change time. They didn't change time, papas. What did they change was the effect of the dim light. In other words, when was the light dim, dimmer, dimmer, dimmest, etc. So a dim electric bulb positioned at varying distances. So if the dim light was close to the eye, it'll appear a little brighter. If the dim light was further away from the eye, it'll appear even dimmer. Okay. Was switched on at 1 minute intervals for a period of 10 seconds. Not all measurements were at different distances from the eye. Whenever they do this, dolls, you must know they're going to have a question on this. Okay. So it would maybe 2 time intervals. It was at the same distance from the eye. How would you know which 2 time intervals? We'll see just now. During this period, the diameter of the pupil was measured. So you can picture it. You got one eye covered, you got the light there, and then you're moving the light further and further away from the eye to make it dimmer and dimmer or whatever. Let's see. Let's see. We don't know if it's further and further. Let's look at it. In time interval 1, the diameter of the pupil was 2. Time interval 2, it was 4. Oh, it got wider. So when does the diameter get bigger? In dim light. So that means in time interval 2, it was dimmer. Time interval 3, even dimmer. Time interval 4, oh, the same as 3. So that means time interval 3 and 4, the light was the same distance from the eye because the pupil was the same size. Time interval 5, oh, it became bigger. That means the light was moved further away. Time interval 6, most probably it's brought a little closer. Time interval 7, even closer because the pupil got smaller. Time interval 8, very, very close because the pupil got very small. Time interval 9, okay, it was a little further away from the eye. So they moved it varying distances. Let's just say varying light intensities. Isn't that so? Varying light intensities will come from the varying distances from the eye. Good. Now let's go down. Darlings, this is a pretty long lesson, and I'm hoping that you can stop it in between and then focus and try and answer a question and then start it again and see whether you got the answers right. Name the mechanism by which the iris regulates the amount of light entering the eye. Of course, the answer there is pupillary mechanism. Identify the independent variable and dependent variable and plot a line graph. Papas, let's see. Can you see that? Distance of the light bulb from the eye. Distance of the light bulb from the eye, because why, babies? What did they say here? They said varying light intensities, did an investigation to determine the effect of dim light. So they got distances of the light from the eye. Look at how they got it, boo-boos. So if you said effect of dim, if you said dim light as your independent variable, you can't say dim light. You can say varying intensities of dim light. or varying light intensities, but they got it as distance of the light bulb from the eye. Something for sure, they didn't put time intervals. Remember I told you that there? Okay? You do not use this as your independent variable. Right, Diane? So always go back. And if you go back and you see here, what did the scientists investigate? And then you'll be able to judge. All right? And then your dependent variable would be my baby's diameter of the pupil. Plot a line graph. I just want to go to the answer, the line graph, babies. You know, just in case you're rushing for time, which you shouldn't be. You should time yourself and manage your time. One way in which you can learn to manage your time is by taking entire papers and sitting and writing them. Once you have learned your work, write it and see whether you can finish the paper in 2.5 hours. Make sure it's a paper you haven't seen before, though. So 1 mark is given for the name of the axis and the scale. This is called the scale. Remember, the lowest number here, highest there, and the difference between any 2 consecutive numbers must be the same. You can't put 0, 1, 5, 7. You can't do something like that, okay? So 1 mark for both of that. 1 mark for time interval and your scale over here. Okay, all right? And then only 2 marks for your plotting, papas. Only 2 marks. And you must put your caption. Diameter of the pupil exposed to varying distances of dim light in 1-minute intervals. You didn't even have to say it in that particular way. So a graph showing the change in the diameter of the pupil when exposed to varying distances of dim light. Okay, so notice the mark scheme. Type of graph, if you drew a line graph, 1 mark. So my darlings, if you had no time to even plot, just put 2 points there and just draw a line through it. You would have got the type of graph right. Some people just do this and don't draw anything, you've lost your type of graph. Okay, title of the graph, that's your caption, 1 mark. Scale for y-axis, are you with me? And with an interval of the bars, we don't have bars here. Scale for y-axis, and it'll be a title. Scale for y-axis, and what's it? I think correct scale, and label for x-axis and label for, so actually we take both that is 1, both that is 1, okay? All right, and plotting. 1 mark for 1 to 8 points plotted correctly. 2 marks for all 9 points. So you had, what would most of you do? Oh my God, this is such a big graph. And it's going to take me so long. And you'll take half an hour to do this here, darlings. When you could have lost 1 mark by just plotting 2 points, left the rest of it, gone to the, and then if you had time, come back to this, you'd have lost 1 mark. So please use your time wisely. Okay, let's go back now. Between which 2 measurements did the largest decrease in the diameter of the pupil occur? Let's go back to the table. Largest decrease. Where did it decrease? It decreased from 7 to 3. It decreased from 8 to 7. It decreased from 3 to 1. There it increased. But from 8 to 7, it was only 1. 7 to 3, it was 4. So which time intervals? Between the 6th and 7th time interval. Explain why the diameter of the pupil remained the same during the 3rd and 4th intervals. Of course, the light was the same distance away from the eye. Therefore, the pupil was the same size. Okay? List 1 way in which scientists ensured validity. 1 way in which they ensured, in other words, they did something over here, papas. What did they do? They, see, it was positioned at varying distances and switch on 1-minute intervals. They only use 1-minute intervals for switching it on. It was switched on only for 10 seconds. Okay. They covered only 1 eye. Explain why the results cannot be considered to be reliable. Reliability is about how many people did this investigation? How big was your sample? Or was there any repetition? So let's look at it, okay? We went through all of that. What happened to our memo, doo-doos? I think we lost our memo somewhere here, babies. But let's answer it ourselves. We don't have to rely on the memo. Let's go back there, babies. Let's go reset. We did that. How they explain. I think we've done all of that there, babies. Why the results cannot be considered to be reliable. Only one person did this. It was not carried out with a huge sample. The greater the number of people in an investigation, the greater the reliability. Okay. Or the investigation was not repeated. So that's about reliability. Good, my darlings. So we've done 2 questions in the eye. Of course, before I move out of here, they could do a similar question which appeared in another paper, and it is in your white revision booklet, where they spoke about the curvature of the lens, you know. So if I had to just draw the lens again, knowing just how fantastically I can draw. So if you had to draw the lens, so if you find from there to there, my darlings, from there to there is the curvature of the lens. So if you find that the distance from there to there gets bigger and bigger, means you're looking at an object that is closer and closer to the eye. Because for near vision, it becomes more and more biconvex. So if you're looking at the curvature of the lens, they're looking at the process of accommodation. For those of you who just want to extend yourself a bit, know that the closer an object is to your eye, the more light comes into your eye as well. So generally you find that closer objects give off more light, you know? And your pupil also tends to get a bit small when your object is closer to your eye. So we generally talk about the convexity of the lens, but should they ask you, know that your pupil can also get small the closer an object is to your eye, because more light comes off the object into your eye. But focus first on the convexity of the lens. So with that being done, I think we have wrapped up your eye. If you do come across questions, lovely, go to them, and should you have some difficulty, there should be some way of contacting your teachers, and I'm sure your teachers would give you a fantastic response to a question that is addling your brain. Okay, now we're going to the ear. The diagram below shows the structure of the human ear. I'd love to give you 1 minute to quickly label from A to G. You can do a mental label. Great. Let's begin. A. What is A, my darlings? That is your auditory canal. Remember, it allows sound waves to pass through and to cause the tympanic membrane to vibrate. It's also got your wax glands, etc. here. B. Now, it's not pointing to your ossicles, isn't that? So these are your hammer, anvils, dirip. It's pointing to this entire area, so that's your middle ear. C, your 3 semicircular canals. D, your cochlea, the snail-like structure. E, your auditory nerve. F, your eustachian tube that opens up into your throat. G, your round window, which prevents echoing in your ear by absorbing the pressure waves set up in the inner ear. It prevents distortion of sound. It helps you to hear very clearly by absorbing the pressure waves set up in the inner ear. Okay, so these 3 semicircular canals are to do with balance. The cochlea is to do with hearing.
Let's see the questions. Label parts A, C, and D. We've done that. Identify the letter representing the auditory nerve. Of course, that will be E. By the way, from here, let's just do a drawing again. Okay, from here, you would find that the impulses would go to your cerebrum to bring about the sensation of hearing. However, a branch from here, my darlings, the vestibular branch from here will go to your cerebellum. Now, vestibular branch of the auditory nerves, but there are certain things you got to learn off by heart. Okay, so this now is your vestibular, so a branch of the auditory nerve called the vestibular branch goes to your cerebellum. So the vestibular branch, sis, my fantastic spelling again. The vestibular branch of the auditory nerve, vestibular branch, you must say of the auditory nerve. The vestibular branch of the auditory nerve goes to the cerebellum. So here you will be able to restore balance once the cerebellum receives impulses via this branch of the auditory nerve. Okay, so this then, my darlings, is the auditory nerve, let's just say. So that is the auditory nerve. So and the auditory nerve will send impulses up to the cerebrum to bring about the sensation of hearing. So that one there is for hearing. Hearing. That is for hearing. And of course there, once the messages go up to the cerebellum will respond by sending impulses to your muscles to restore balance. So please know that, my darlings, know that very clearly. Okay? I'm trying to be as explicit as possible so you will have no confusion. Explain what type of neuron makes up that auditory nerve. Remember, the auditory nerve takes impulses only upwards. No impulses come from the cerebellum or the cerebrum down to the ear. No, not at all. So because impulses only go upwards, and the neurons that take impulses up to the central nervous system, my dear children, are your sensory neurons. So what type of neurons make up this auditory nerve? Sensory neurons. Okay? Even the optic nerve is sensory neurons. Explain the significance of label C being arranged in different planes. Different planes, we actually say they are arranged at right angles to each other. So that when the fluid moves inside here, no? Oh, yeah. By the way, you need fluid to move for receptors to be stimulated. What do you call the receptors inside each of these areas? Of course, here in your semicircular canals, the receptors over here, my darlings, the receptors over here are called your cristae. Those are your cristae. They are sensitive to the speed and direction of movement. And the receptors that lie inside over here, not on the surface as I'm drawing it, that is your organ of Corti. Let me just put in the text over there so you know that that is your cristae, and they are sensitive to the speed and direction of movement. And of course, this one here is your organ of Corti. Organ of Corti is so microscopic, darling. When you think organ of Corti, you think something as big as the heart. God knows why they call it organ of Corti. So that is now your organ of Corti. This is sensitive to your pressure waves set up in the inner ear. This is sensitive to the speed and direction of movement. And there's one more that is found here in your succulus and utriculus, which lie inside of there. And what are they called, my darlings? The one that is lying inside the succulus and utriculus that is sensitive to the position of your head is maculae.
So what I've drawn is 3 receptors, your maculae, which lie there, your cristae, which lie in the semicircular canal, the organ of Corti, which lies in your cochlea. Just as a recap. So important to do recap, my darlings. And as I said to my pupils, know your theory first. No matter how many questions you go through, if you don't know your theory solidly, you're going to falter in your paper. So, explain the significance for 2 marks. So they lie at right angles to each other in 3 different planes, so that when the endolymph, the fluid that moves here, moves in any one of the semicircular canals, you can pick up the speed and direction of the movement because you got cristae in each one of them. So because they are arranged at a change in the speed or direction of movement in any plane can be detected in order to maintain balance. So no matter which direction, whichever speed and direction of movement there is, because they arrange in 3 different planes, one of them would be able to detect them. At least one of them would be able to detect them, that change or that stimulus, to restore your balance. When a person is close to a bomb explosion, a significant amount of pressure is exerted on the ear. Explain the processes we'll have on hearing, 3 marks. So the pressure is exerted from here. No, the pinna, the airwaves will go through here and exert the pressure there on your tympanic membrane. In other words, your tympanic membrane will not be able to vibrate freely, isn't that so? Or your tympanic membrane may rupture. So in other words, they're asking you the function of the tympanic membrane in this question. So less vibrations or maybe no vibrations will be transmitted to the oval window or the ossicles. So here, the ossicles, and that is your oval window. And thus you'd find that the cochlea will not be stimulated. I'd like to say the organ of Corti will be stimulated less, leading to hearing loss. I would say the organ of Corti will not be stimulated, leading to a loss in hearing. In other words, the person would be deaf if your tympanic membrane ruptures. But if you have it just a temporary situation where that air pressure is pressing down on your tympanic membrane, darlings, then you would find that it will not vibrate properly because you need equal pressure on either sides of your tympanic membrane. So here and here, the pressure must be equal. So the tympanic membrane will vibrate freely. If it doesn't vibrate freely, then once again, notice how they say it, less vibrations will be transferred to the oval window and the organ of Corti will not be stimulated or stimulated less. So if you say it will not vibrated properly, then you will say less vibrations, the organ of Corti will be stimulated less, leading to hearing loss or hearing will be impaired. If you say the tympanic membrane was ruptured, then the tympanic membrane will not vibrate properly. Sure, no vibrations will be transmitted because it's ruptured. No vibrations. The cochlea will, or the organ of Corti will not be stimulated, leading to hearing loss. So let's be very specific. Okay, this one does not answer as properly as it should be answered. Are you okay? So in other words, it's just asking you, do you know the function of the tympanic membrane? Because the function is to vibrate, to transmit the vibrations to the ossicles, and then the ossicles will transmit the vibrations to the oval window, and then you'd find that your inner ear vibrations will be set up. So if you don't transmit the vibrations to this oval window, then no vibrations will be set up in the inner ear. Organ of Corti will not be stimulated, impulses will not be created and taken by the auditory nerve to the cerebrum, and therefore there will be no hearing. So while they gave this as a memo, darlings, sometimes they would want you to say the organ of Corti will not be stimulated, impulses will not be created, and therefore no impulse will be carried by the auditory nerve to the cerebrum, and therefore you lose the sensation of hearing. So whilst this may be the memo for this time, as I said to you, just be on your guard. I don't want to pause my sharing. Sorry about this, Nunos. How do I unpause this? Resume share. Oh, sorry about that, babies. Sorry about that, my darlings. So here we go. So while this may be the memo for this particular answer, cover your bases. Don't do what you do in English. Forgive me. I've seen children writing for half a page for 2 marks. And of course, you know what you are doing, but we do not require that for life sciences. It's a very specific language. Be specific when you are answering it as well. But if you find there's a mark scheme like 3, 4, and 5, try and cover your bases, because if they say explain, if they say explain, they will have to read what you were saying. They got to hear what you were saying, because they didn't say state 2 facts or state 2 functions. Okay, so bear that in mind. I see I have actually changed the order of the questions somehow. Okay. I saw a question here which included quite a few things, so I said, let me go through it. Read the following extract about motor neuron disease. Now, don't you dare do this in your paper. Oh my God, ma'am did not teach me about motor neuron disease. If there's something I haven't or your teacher hasn't taught you about, my darlings, it's because it's not in your text. And if it does appear in your paper, they would give you information about it. Here, motor neuron diseases refer to a group of conditions that cause the motor neurons in the nerves of the spinal cord and brain to progressively lose their function. Remember, motor neurons carry impulses down to your effectors, and your effectors will now bring about an appropriate response. So if you find that your motor neurons are being damaged, my darlings, that means impulses are not going to be taken to your effectors, and thus you'll not be able to carry out certain reactions. Motor neuron diseases are rare but serious and incurable forms of progressive neurodegeneration. That's nerve degeneration, your nerves, like dying, simply put. Motor neurons are nerve cells that send impulses to the muscles. Genetic, viral, and environmental issues may play a role, so those become the causes. Motor neuron disease can appear at any age, but most patients are over 40. It affects men more than women. According to the extract, what is the definition of motor neuron disease? Now, this is a straightforward question. There's it. A group of conditions that causes the motor neurons in the nerves of the spinal cord and brain to progressively lose the function. From the extract, 2 factors, there we go, genetic, viral, environmental. Explain the possible effect that this disease will have on a reflex action if a person was to step on a thorn. Oh, that is okay, isn't that so? Because we know if the motor neuron is damaged, that means no impulse will go down to the muscle. Your muscle will not contract and move your foot away from the thorn, and you would find that you would find damage, but you'd be able to feel the pain. Exactly what we came across before. The person would be able to feel the pain? No, because the sensory neuron is not damaged. Yeah, and the impulse will go up to the brain so you can feel the pain, but you cannot react because the motor neuron is not functioning, therefore the muscle will not be stimulated. Now look at how strict this memo was. I would think that's awful for a child not to give the child leeway, because I would not say, therefore the muscle would not be stimulated. I would say, I'm sure they would have allowed you, Papa. Therefore the muscle would not contract and you would not pull your foot away from the thorn, and you could be hurt by the thorn. You would be hurt by the thorn. Possible effect. Why didn't you say that the thorn would poke into you or you'd have extreme injury? No mark was accorded to that. Okay, so this becomes one of the thorny issues in life sciences. Draw a label diagram of the motor neuron. Please, loves, you must be able to draw the motor neuron. You must be able to do the sensory neuron. 4 marks. Sometimes they give it to you 5 marks. Notice they didn't give you a caption over here. Always put diagram of the motor neuron. Here they didn't give you a mark for the caption because I think they told you draw the motor neuron. But 3 labels. See, 3. Know at least 3 labels, I always say. Know at least 3 labels. Dendrites, nucleus, etc. Okay, let's go back here. One of the defects of the eye is astigmatism. Remember, 4 defects of the eye. Short-sightedness, corrected with convex or biconvex lenses. Long-sightedness, biconcave or concave lenses. Long-sightedness, convex or biconvex lenses. Okay. Cataract surgery. Astigmatism is when your cornea is not smoothly rounded. I don't know what actually causes it, but they don't want to know the cause. They want to know, like, what is the reason? Isn't that so, my darlings? So it is caused by the irregular curvature of the front surface of the cornea. Is when your cornea is not evenly rounded. What would you use? Now here they got prescription lenses, but yet in another text, another memo, they said use ungrounded lenses. Not ungrounded lenses. Yeah, use ungrounded lenses. Prescription lenses, ungrounded. Is it ungrounded? No, unevenly grounded. Sorry, Madai. Unevenly grounded lenses in your glasses. Unevenly grounded lenses in your glasses. Because your cornea is uneven, you use unevenly grounded lenses. So here they just put prescription lenses as the answer. But yet in another memo, you had to put unevenly grounded lenses. I would suggest you go with unevenly grounded lenses. Name 2 defects of the eye associated with lenses: short-sightedness, long-sightedness, cataracts. Okay, darlings, let's go to one more question before we wrap up this very long session that we had. Please, you must know the pathway taken by sound waves to bring about hearing, and you must know balance of by heart. Notice this particular diagram shows you all the parts of the inner ear. These are your 3 semicircular canals. Babies. Your 3 semicircular canals. That's 1, 2, and 3. And of course, here you'd have your cristae. This part here, you'd have your utriculus and sacculus. And there you'd have your second set of receptors for balance called your maculae. So these parts here and that part is to do with balance. Here, your organ of Corti is to do with hearing. Your cochlea, this is your cochlea, and inside it, you got your organ of Corti. Remember, all these receptors, cristae, maculae, and organ of Corti are only stimulated when a fluid called endolymph moves. Only when endolymph moves is it stimulated. Just remember that in case they ask you in a question. Endolymph. Great. Okay, good. Only when the endolymph moves are receptors stimulated. So this is for speed and direction. Remember, they're in 3 different planes so that no matter which direction your head is moved or in whichever direction the movement occurs, at least one of these semicircular canals would be stimulated. What will be stimulated? The cristae in one of the semicircular canals will be stimulated, impulses will be created, and balance will be restored. By the way, where would the impulses go to from here? From these things and these things up to the cerebellum. From here up to the cerebrum. To identify structures 1 and 2, semicircular canals, cochlea. Explain the significance of structure labeled 2 being coiled. We always say this is a snail-like structure. And we've always learned in biology if something is coiled or long or convoluted, it always increases surface area. So it increases the surface area of the inner ear for the organ of Corti, I'd say, thereby helping to boost low frequency sounds. allowing you to hear soft sounds. So it increases the surface area of the inner ear, boosting low frequency sounds, allowing us to hear soft sounds. If you can't digest that, look at it about 5 times over and learn it off by heart. And here's the question. Describe the role of the structure above in maintaining balance. As I said to you, this is the memo I'd like for you to learn off by heart. A change in speed and direction of movement stimulates the cristae in the semicircular canals. The stimulus is converted to an impulse. Impulse is transmitted to the cerebellum via the auditory nerve, or we learned is via the vestibular branch of the auditory nerve. The cerebellum sends impulses to the muscles to restore balance. Notice they didn't speak about the sacculus and utriculus because they only referred to the semicircular canals. And in some cases, they ask you for balance regarding the semicircular canals and the cristae. Are you with me? So there you got to refer to both. This is a lovely question. I just want to go through it for the sake of the pie chart. Please do not go into an exam and say, oh, I didn't think the pie chart was going to be in the paper. I don't have a protractor. Before you step into a life science paper, you must have the protractor. You must have the compass. Good. I couldn't get that word. The table below indicates the percentage of visually impaired people in the world suffering from different visual defects. For example, me, I am short-sighted, so I'd be there in the 30% of the people. Blindness, 2%, others, whatever, okay? Which visual defect is the most common amongst the world population? Of course, is long-sightedness. They can see things far away, but they can't see things close. Ooh, I'm not common. In some cases where people are blind, the condition is caused by cataracts. Explain why people with cataracts may become blind. You must know that cataracts is where your lens becomes cloudy and prevents the light rays from going through. It also prevents the refraction of the light rays so they don't meet on the yellow spot of the retina. Are you with me? And therefore, no image will be created and no impulse will be sent up to the cerebrum. You will not see. State one way in which cataracts can be treated, of course, surgery. Explain why long-sighted people need to wear glasses with biconvex lenses as a corrective measure. Because long-sighted people, what happens with them, darling? The lens fails to become more biconvex. And therefore, they cannot refract the light rays more greatly. And therefore, they will have to wear your biconvex lenses, which helps you to refract the light rays so that they meet on the retina. Just now we look at the answers to see how we got the 4 marks, babies. Name a visual defect that is characterized by uneven cornea or lens. Astigmatism. Draw a pie chart. I'm going to come back to that. So let's go back to the answer. Explain why people with cataracts become blind. The lens becomes opaque or cloudy. Therefore, light cannot pass through. That's all they wanted, surgery. The lens is less convex or the eyeball is too short. Remember we said to you? This causes the light rays to fall behind the retina. Wow, look at that there. Look at what they included. Even I didn't include that. I would have said the lens fails to become more biconvex. A biconvex lens, do you correct it? A biconvex lens increases the refractive power. Therefore, light rays are focused on the retina. Look at how tight this memo was. Look at how tight. So this causes the light rays to fall behind the retina. I love this because remember I told you with long-sightedness, the eyeball is shorter than normal. So the light rays fall behind the retina. With short-sightedness, they fall in front of the retina because the eyeball is too long. Here, your eyeball is short, the light rays fall behind the retina. Isn't that beautiful, my darlings? So here they included both things. Both of the defects with long-sightedness. The 2 defects with long-sightedness is that your lens fails to become more biconvex. And secondly, your eyeball is shorter than normal. that included both of that in your answer. All right? Astigmatism, etc. Now, when it comes to your pie chart, first of all, you must show your calculations. They showed you the percentage of people who are blind. So work out how many degrees. So 2 out of the full percentage, 100 times 360 or 1. Show this because normally marks are given. And how many marks are given for that, darlings? Let's see. 2 marks for all 4 correct. Don't leave this out. Okay? 1 mark for drawing your pie chart. Another mark for your caption. So, pie chart 1. Title 1. So without even drawing your pie chart, you got 4 marks, nanas. So what you could have drawn is just drawing a circle. And then if you got time, if you're rushing for time, then go and put in all of this here using your protractor. How's that? Without drawing all these here, you'd have got 4 marks. But you don't want to lose any mark at all, isn't it so, darlings? So notice they labeled each piece of your pie. Great. I think we're doing very well. I just want to do one more on the rate at which you react when you have alcohol. I'm going to go through that, okay? That's a simple, there we go. This one was about stamina. It was a lovely question for investigation. There's also another one on measuring how quickly you react to a stimulus. And you find that the more often you are exposed to a stimulus, the faster is your reaction time. It's like almost training yourself to react. And then after a while, you just become tired and your reaction time becomes slower. Okay? If I do find the question, most probably I will just do a little video on that. Let's wrap up this session. It's been a long session. A scientific investigation was conducted to 20 cyclists, very low reliability here, isn't that so? You need a large number for reliability. 20 is not a large number. A scientific investigation was conducted to 20 cyclists to determine whether caffeine can increase stamina. Here you go. You'll be able to work out what is the independent and dependent variable, whether caffeine can increase stamina. So your increase in stamina would be the results you're looking for. But how are you going to measure it? How would you know if stamina increased? It must be by some activity which is done, you know. So we'll see how they will measure stamina just now. It's like rate of photosynthesis. How are you going to see what was the rate of photosynthesis? There must be a way in which you measure it. Isn't that so, darlings? And whether caffeine, so the absence or presence of caffeine would be the independent variable, whether you had caffeine or you didn't have caffeine. Procedure was as follows. A group of cyclists was given the first cup of coffee containing no caffeine to drink. But they were not told about it, darlings. They were just given a cup of coffee. So you assuming you're having a cup of coffee, you'd assume because coffee is a caffeinated drink, it'd have caffeine. A group was then asked to cycle for as long as possible at the same speed. Just cycle. So stamina is measured by how much of endurance you got. How long can you do a particular activity? I would never want to put myself through this because even climbing up a flight of stairs was so difficult. They then cycled around a 400-meter track on level ground. And they wanted to see how long can they cycle. So that is the way in which they measured their stamina. Okay. The group was given a second cup of coffee thereafter, containing caffeine to drink before cycling for the second time. So they used the same group of cycles. Reason, you want to control your variables. You can't use one group of cyclists and then use another 20 people for another group, my darlings, because there'll be far too many variables. However, between one activity and the other, you got to give them ample time to relax. I would preferably have done that on one day, and on another day, maybe 2 days later, given them that second cup of coffee. Are you with me? And then taken my results. Because if you don't allow them to rest in between, then you're interfering with the validity of your investigation. The table below shows the average time the group of cyclists was able to pedal after drinking decaffeinated coffee, coffee without caffeine, and drinking coffee with caffeine. So the type of coffee would be the independent variable. The average duration of cycling would be the dependent variable. Are you with me? Because this is how you measure your stamina. State the question that the scientists were trying to answer, what we're trying to investigate, whether caffeine can increase your stamina. Identify the independent variable, dependent variable, and how, there you go, loves. Dependent variable and how it was measured. Dependent variable, if you look here, boo-boos, you would have worked out your independent and dependent. So the dependent one is stamina. How did you measure it? By measuring the average duration of cycling. Average is very important, isn't it? So, darlings, okay, average would be more reliability. What is the relationship between caffeine consumption and the stamina? Of course, coffee increases the average duration of cycling. Okay. Explain the purpose of giving the cyclist decaffeinated coffee to drink during the investigation, because that would serve as your control investigation, so that you can now be able to verify that it was coffee with caffeine that made a difference and nothing else. Because there's only one thing different between that group and this group, was that they didn't have caffeine, and then later on they had caffeine. So the purpose would be to serve as a control, something to compare with. We look at the memo just now. The cyclists were not told whether or not the coffee they drank contained caffeine. Explain why the scientists were not given this information. Of course, psychologically, this would affect their performance, and this will now interfere with the validity of the investigation. Because if I'm telling you, I'm giving you an energy drink, automatically you will think you got a lot of energy, and even though it was not an energy drink, you will make sure it manifests as something that shows that you have a lot of energy. The procedure does not include how much time passed between each cycling exercise. Remember I told you this? Explain why this is an important factor to know. You should be able to predict all of this, my darlings. Now let's look at the memo. Does drinking coffee containing caffeine increase stamina? Independent factor, amount of caffeine or presence or absence of caffeine. Stamina was a dependent. How did you measure? The average duration of cycling. The average cycling time of the cyclist increased with the use of caffeine. You wanted to know the relationship. Why would you have that decaffeinated coffee? Serves as a control to eliminate any other factor that may cause an increase in stamina. So you know for sure that it was the caffeine that caused the change in stamina and nothing else. To eliminate any other factor. Notice where they gave that mark. To eliminate any other factor that may cause an increase in stamina. Because there was only one difference between the control group and what we call your experimental group, so that you can prove that no other factor had caused an increase in stamina. I'm sure they would have given you a mark that way as well. So why is it they were not told that they had no caffeine? Knowing whether caffeine is taken or not may subconsciously influence their performance. 2 marks. Look at how they gave that mark. Oh my goodness, you know. Or the participants may think they have more stamina if they know they are taking caffeine, and this may influence their performance. So why did you have to have, what did they say, darlings? Something that we had contemplated when we were thinking about investigations and how they must be conducted. The procedure does not indicate how much of time passed. Why is it important? 2 marks. If too little time passes between exercise tests, the participants may be tired, which will influence their stamina and therefore the validity. Are you with me? Or you could have said the participants must be equally rested for both tests to ensure that, always to ensure validity. Or the cyclists may perform better in the second test because they are better warmed up. This may also influence validity. Whichever one you chose, you gotta say to ensure validity. With that, my darlings, I think we covered a fair variety of questions. I do hope this makes a difference to how you answer questions and how you are mindful of mark schemes. And for you to remember, if they're using the word explain or what will be the consequences of or predict, they're always asking you the function of a part in a different way. So do not be worried. Learn your theory, and should you learn your theory very well, you'd be able to answer any of these questions. In the next video, I'll be talking about chemical coordination. Until then, my darlings, God bless.